One of the most important clinical skills in hematology is distinguishing iron deficiency anemia from other conditions that produce a similar blood picture: small, pale red cells in the setting of anemia. The three major conditions that can look like iron deficiency on a blood count are anemia of chronic disease, beta thalassemia minor, and sideroblastic anemia. Each has a distinct laboratory fingerprint, and knowing how to read that fingerprint is essential. Starting with anemia of chronic disease. In iron deficiency, serum iron is low and TIBC is high. The bus analogy again: few passengers, lots of empty seats. In anemia of chronic disease, serum iron is also low, but here is the key: TIBC is also low. The body is not trying to capture more iron. Instead, it is deliberately sequestering iron as part of an inflammatory defense mechanism. The theory being that by locking iron away, the body deprives bacteria of a nutrient they need to replicate. So both iron and TIBC go down together. The single distinction separates these two conditions and is a high-yield clinical point. Next, beta thalassemia minor. This is a genetic condition affecting globin chain production. Interestingly, the iron studies in beta thalassemia minor may be completely normal. The distinguishing laboratory clue is found on hemoglobin electrophoresis, specifically an elevated hemoglobin A2 fraction. Hemoglobin A2 is an alternative form of hemoglobin made up of alpha chains and delta chains. When the beta-talta chains is impaired, the body compensates by upregulating delta chain production, which pairs with available alpha chains to form more hemoglobin A2. An elevated A2 on electrophoresis in the right clinical context clinches the diagnosis. The clue is in the hemoglobin pattern, not the iron panel. Finally, sideroblastic anemia. This is a fundamentally different category. Rather than a deficiency of iron, this is a condition where iron cannot be properly incorporated into heme during red cell development. The result is that iron accumulates inside developing red cell precursors in the bone marrow. Specifically, it accumulates in the mitochondria of those cells. This location matters because heme synthesis itself occurs partly within mitochondria, so iron gets trapped at the very site where it was supposed to be used. When these cells are stained on bone marrow examination, the iron deposits form a ring around the nucleus, hence the term ring sideroblast. The mitochondrial location of the iron is not just a pathological curiosity. It reflects the mechanistic failure at the heart of the disease. Sideroblastic anemia can be either inherited or acquired. The inherited form follows an X-linked pattern. The acquired forms have several causes: myelodysplastic syndromes, excessive alcohol use, vitamin B6 deficiency, and lead poisoning.